Okay, so let's get started here. I'd like to welcome you all to the session entitled What is the Real Molecular Target of Metformin? My name is Lawrence Kazak. I'm an associate professor at McGill University. We have a great selection of speakers today. And so first up is Barbara Nikolajczyk. Barbara is a professor of pharmacology and nutritional sciences at the University of Kentucky, and her research focuses on identifying the role that immune cells play in human obesity and aging, and type 2 diabetes. So, welcome.
All right. So I want to thank the organizers for inviting me to present my team's work today on metformin and inflammation, and I'll be focusing on cellular mechanisms rather than molecular mechanisms, which I will leave up to Nav. So I am going to begin today by introducing you as to why we're focusing our work on one particular immune cell subset, that is CD4 T cells, as the dominant influencer of inflammation in obesity, diabetes, and actually aging. And then I'll be showing you some data from in vitro utilization of metformin and what that does to improve T cell function before I'll be moving into unpublished data on outcomes from a clinical trial with a metformin intervention. All right, so why CD4 T cells? So the way that we look at this in our in vitro analysis is what I call a baseline experiment, where we measure something called cytokine competency. This doesn't tell you necessarily what the cells are doing inside the people, because that is not really possible to do, but it tells you what the cells are able to do ex vivo. So we take purified peripheral blood mononuclear cells, or PBMCs, or alternatively, we'll use CD4s for some studies. We pan-stimulate them through the T cell receptor, or in the PBMC studies, we'll also use LPS to activate primarily the myeloid cells. And then we measure a bevy of cytokines after about 24 to 48 hours as our measure of function. We then put all these data points together in a mathematical modeling approach called partial least square discriminant analysis, where each compendium point from the analysis gives you inflammation for that particular sample. So when we did this with over 7,000 data points that were produced by cells from people with diabetes in red, or without diabetes but BMI matched in blue, we can see here sharp separation in the overall clouds of inflammation generated by by PBMCs from these two groups. And when we ask which amongst the 7,000 and some points were most important for identifying type 2 diabetes inflammation, we can see here really starting, and this is the most important one is at the bottom, the 10th most important one is at the top, and then you go up to 7,000, but I don't want to show you that today. We see here that the majority of those most important cytokines for defining type 2 diabetes inflammation are cytokines made predominantly by this one T cell subset, CD4 T cells, and that's amongst all the cells in PBMCs. All right, so we leveraged this into studies that we were really starting in an aging scenario, and we were just a little bit lazy on this, so we did our baseline experiment on either faculty on our floor who were average age of about 60, or postdocs on the floor with an average age of about 30. So this is really a highly educated cohort. We focused on CD4s based on that previous data, and perhaps you're not surprised that there is a significant difference in the inflammatory cloud produced by the cells from the older folks versus the cells from the younger folks. And if we again look at our model and ask what are the most important cytokines for differentiating this age-related inflammation or inflammaging, you can see here highlighted in red that the vast majority of those cytokines that we measured were either sort of the stereotypic Th17 cytokines, Th17s being a subset of CD4s, and those included things like IL-17A and IL-17F, or Th17 supportive cytokines like IL-6. So we defined inflammaging in this faculty cohort as a Th17 profile. But I don't want, especially you trainees, to go back home and wag your finger at your faculty saying, oh my gosh, you guys are inflammaging and falling apart. To indicate that, we partnered with our Alzheimer's Disease Center and looked at the same Th17 inflammaging signature in people who were either cognitively healthy down to people who had mild dementia. And we can see here a negative association with that Th17 signature, regardless of whether we looked at things of both sexes, but with this signature especially being strong in the older females. And these folks were not terribly old. They were like 74 with a BMI of about 26. So what this tells us is what Luigi Ferrucci discovered 20 years ago at the NIA, and that is that inflammation may not be all bad news, so give your older faculty a little bit of a break. All right, so when we showed these data in our faculty research in progress, Phil Kerr, my clinical coordinator, said, Barb, that's a ridiculous cohort. There aren't so many PhDs in the United States. What if we look at a cohort that is more representative of people in America today? So we recruited people who hovered around the classic obese BMI of 30, this similar age difference, so 60-year-olds and 30-year-olds, and then people from both genders, all of whom were non-diabetes as indicated by a hemoglobin A1C of less than 6.5. We did that same baseline experiment with the cells from these people, and you can see here a sharp separation in obesity-associated inflammation. But what's interesting here is that if anything, you lack Th17 cytokines, especially IL-17F and IL-17A, differentiating inflammation in these two age groups, telling us that inflammation is not defined by a Th17 profile in normal glycemic people with obesity. It's really more a mixed CD4 T cell profile, remembering that in this study, we limited the analysis to CD4s. So really trying to understand some cellular mechanisms that may describe these differences in inflammation. We took advantage of a seahorse mitochondrial stress test where we see very high mitochondrial OXPHOS as indicated by oxygen consumption rate in this particular assay, and also an increase in mitochondrial compared to non-mitochondrial respiration in this ratio analysis in the cells from the lean, healthy postdocs and professors. If we did the same experiment in a more general American cohort, people with obesity, we see here really little, if any, difference in the mitochondrial bioenergetics comparing cells from older versus younger people with obesity. And in fact, this top mitochondrial respiration is really similar to what we see in the younger, lean people, telling us that inflammation correlates with higher mitochondrial respiration, but only in CD4 T cells from lean individuals. So we asked whether these kind of mechanistic analyses might be something that we need to take into consideration when we were developing our investigations of metformin as a kind of geroprotective strategy to mitigate age-related health problems. And so we first attacked this in vitro, where we added metformin to this baseline experiment, activating cells from our older lean cohort. And you can see here, perhaps unsurprising, really strong anti-inflammatory functions of metformin. It knocked down this Th17 profile comprehensively. What happened when we did the same experiment in the cells from people with obesity? We see here, in the yellowish color, that's with the addition of metformin. Black is a vehicle control. And we see here a relatively low accuracy model. Our bioinformatics colleagues are looking for an accuracy of about 70% for a predictive model, telling us that metformin has little, if any, effect on this obesity-associated inflammation profile. We then put our thumb on the scale by getting rid of all the cytokines that were really kind of the noise part of this analysis, and we really didn't improve the model all that much. It just barely tipped it over that 70% cut point. So perhaps our univariate analysis, where we showed that metformin blocks production of IL-9 in this particular obesity scenario, is as good as it's going to get, right? We didn't get any bonus from the more complex analysis that takes advantage of known and unknown interactions between the cytokines. Concluding really that metformin in vitro is modestly lowering inflammation from older subjects with obesity. So really looking back on that mitochondrial bioenergetics that I showed you, one of the major mechanisms by which mitochondria are regulated, especially in aging, is through autophagy. And since metformin is known to be an autophagy activator in other contexts, we asked whether this would be similar either in lean or obesity-associated contexts. And to just kind of outline what we looked at this, we've done many different autophagy studies and also some mitophagy studies, and you'll know that there are scads and scads of ways you can measure mitophagy, but we've kind of kept it simple, right? We're working all on primary human cells, really looking at defects in autophagy based on lower LC3B, which is down here on the autophagosome, and accumulation of P62 in the older cells, really telling us that there are defects in autophagy in these T cells from the older people that are repaired by our baseline experiment stimulating the cells in the presence of metformin. We use complementary confocal approaches, which are really looking at LC3 puncta in this autophagosome structure, and we see here again defects in our older compared to our younger cells that are restored specifically in the older cells by metformin. If we did that same experiment in cells from our people with obesity, though, we got really no effect of metformin whatsoever if you compare the gold dots to the black dots in either cells from the younger obese cohort or the older obese cohort. And we can talk about this higher indicator of autophagolysosome formation later on if you're interested. All right, so that's all in vitro. What on earth does it mean? To understand that, we partnered with Benjamin Miller at Oklahoma Medical Research Foundation and Adam Konopka at the University of Wisconsin, who were doing this ANTHEM study where they were taking from a geroprotective perspective people who were middle-aged and older, metabolically characterizing them, and then putting them on metformin. These guys are muscle guys, so they measure muscle mitochondria and other muscle stuff. And honestly, we have mitochondrial envy over these guys because muscle has so many more mitochondria than T cells do. But talking to Benjamin at the age meeting, we saw that this was his cohort. They're right on the line of obesity, such that his people who were basically obese and insulin sensitive were probably very similar to the cohort that we were looking at in Kentucky with our in vitro samples. So we wrote an ancillary project to their anthem, calling it iAnthem, to add tubes of blood as well as indicators of metabolism and autophagy in the immune cells and looking at inflammatory profiling. So if you could put your cameras away, please. This is unpublished data. Thank you very much. So what was really interesting is that when we looked at cells specifically from the insulin-sensitive people, so similar to what we had seen in Kentucky, we see here a low accuracy model if we compare cytokines produced pre-metformin in blue versus post-metformin, and this echoed exactly what we saw in the data with the in vitro metformin intervention. However, quite interestingly, when we expanded this to insulin-resistant subjects whose cells we did not look at in vitro, we got quite a different story. We got sharp separation of the pre- and post-metformin inflammatory clouds with a highly predictive model. And then when we asked which cytokines metformin specifically mitigated and especially lowered in this context, we see more or less a pretty strong Th17 signature, including Th17 support of IL-6 as well as IL-21 and IL-17F, telling us that metformin intervention, so this is a clinical trial, remember, reduces cytokine production by CD4 T cells, but specifically from people who are insulin resistant with obesity. All right, no effect of placebo. Looking into autophagy as a particular, as one way that we might be seeing the differences in mitochondria I showed you in the bioenergetic analyses earlier, we again turn to confocal, looking at the markers that I had described earlier. And if I could focus your attention on the first and second bars, you can see that in our insulin-resistant placebo samples, we have defects in autophagy as compared to relatively low lipidated LC3 in blue compared to the relatively high marker in the insulin-sensitive placebo-treated cells. Also defects looking at LAMP1, looking at co-localization of these, so autophagolysosome formation, and then also accumulation of P62. And if you look carefully at the stars, the only thing that has changed in the metformin intervention, so comparing the second bar to the fourth bar, is when we compare insulin-resistant cells from placebo-treated people to insulin-resistant cells from metformin-treated people. There's absolutely no impact of metformin on these cells from the insulin-sensitive people, telling us that stratifying your metformin outcomes by metabolic status of the patients may yield interesting insights. All right, and also recapitulating our in vitro analyses that were done with our obese but insulin-sensitive people in Kentucky. All right, so now we're moving on to really focus on lysosomes as perhaps the key cellular process that appears to be problematic in insulin resistance that might be mitigated by metformin, really focusing on this protein CISH, which is cytokine-inducible SH2 containing protein, because we see in preliminary analysis CD4 T cells from the insulin-resistant people have less of this critical lysosome regulator than cells from people who have insulin sensitivity. All right, so what I've shown you today is, first of all, one immune cell subset. The Th17s of CD4s are dominating peripheral inflammation in obesity-associated type 2 diabetes and healthy aging, but this may not be all bad given the cognitive decline data that I showed you. I showed you that obesity in the context of insulin sensitivity really is profoundly changing CD4 T cell mitochondria, but through mechanisms that are more or less metformin resistant. In contrast, if we look at cells from people with insulin resistance, metformin intervention, and this was both the clinical trial, well, this was just the clinical trial, we don't have the in vitro analysis of this, is really fundamentally impacting CD4 T cell cytokine profiles as well as CD4 T cell autophagy as potentially the underlying mechanism. With the final take-home being that insulin resistance in the context of obesity really is changing CD4 T cell biology, as indicated by responses to metformin, with our using metformin as a probe, but thinking perhaps as being more stratifying of your individuals when you're thinking about metformin as a geroprotective. And if you want more information on this, my lab manager, Dr. Ida Javidan, is giving a poster on this tomorrow. All right, so I want to say thanks to the folks who did this. Sarah and Lena defined these Th17 signatures in aging and the non-Th17 in obesity. Gabby really highlighted CD4 T cells as major indicators of an inflammatory generating profile in type 2 diabetes. These folks are in our Alzheimer's Disease Center. Jenny did the ADC studies. And then our clinical team consisting of Dr. Phil Kern, who will look up any medical record question I have within about 10 seconds of me texting him. Our analytical teams here. And then finally, Ida, who is really taking on this clinical trial and bringing those results to fruition. So I'll stop here and we will save questions till all of us have spoken.
Perfect. Thanks for staying on time. That was great. Next up, we have Navdeep Chandel, who's going to talk about the molecular mechanisms of metformin action on mitochondria. And Nav is a professor of medicine, biochemistry, and molecular genetics at Northwestern University, and his lab studies how mitochondria control physiology and disease beyond ATP. So,
this thing. Okay, great. So thanks for the nice introduction. I want to thank Paul and Katrine for giving me the opportunity, and as well as the ADA. As Lawrence just told you, I don't work really on anything related to diabetes. I know quite a bit about mitochondria just because I've done it for a long time. And we've sort of stumbled into, this old paradigm, whether metformin hits mitochondrial complex 1. And let me show you some of our genetic data that maybe perhaps points to this. Well, the most important slide, this is why I put it in the beginning, because the people who've done this work, in particular, Zach Zebo, who will be on the job market this fall, I'll present his unpublished data in the second half, and some previous work from Colleen Rezek and other people in the lab, and obviously I've got some fantastic collaborators. Okay, so I think many of us are interested in this question, which is what are the physiological targets of oral administration metformin? As you can see, I've underlined the word oral because I think you could probe the mechanism many ways, but we always try to give, at least in our mice studies, oral rather than IV or IP. Not those other methods cannot be informative, but I think it's important to just upfront say how we've done our studies, which is using oral. And then the other question is when thinking about whatever the targets are, is to think about this particular idea, which is that whatever the target might be, and metformin binds to it, you got to make a refractory version of that target so it doesn't bind, but yet it maintains its catalytic activity. And I would say that Someone can correct me later, but I haven't really seen that kind of stringent test for metformin. Figure out the target, it mutated in a way, doesn't bind to the target, metformin doesn't, but it's still catalytically active. By the way, I'm going to show you stuff around mitochondrial complex 1 where we don't quite hit that ourselves. So full disclosure, to be continued. So, obviously, mitochondrial complex 1 has been around for a long time as a putative mechanism. It reversibly inhibits mitochondrial complex 1, and then it pops off. So the key here is it's reversible. Judy Hurst, who's arguably one of the best people thinking about structure function of mitochondrial complex 1, she's figured out a lot of beautiful work on complex 1, in particular where it binds. And so this beautiful structure is 45 subunits. It comes from 2 different genomes, and there's a couple of sites and a couple of subunits that have been identified. So the proper experiment, by my own criteria, would be to go in and mutate those sites. So now the complex 1 maintains its boot-like structure, but it just doesn't bind to metformin. A tall order, we have not been able to accomplish that. But what we've noticed is the following. So here's mitochondrial complex 1, and it proton pumps, it regenerates NAD, it passes on electrons to the next complex, which is in the electron transport chain. If you knock it out, there's a way you can complement the loss of this, at least in cell culture, which is you can overexpress a yeast protein called NDI1. Tako Yagi's lab did this years ago. I remember just when I was finishing my postdoc, I really enjoyed reading this paper. What they did is they basically put rotenone, a complex 1 inhibitor, in cells. If they overexpress NDI1, it would rescue respiration, NAD regeneration, pass 2 electrons to the next electron transport chain. So you can imagine you can overexpress NDI1 in any given cell, and what you'll get is metformin, if it inhibits complex 1, if NDI1 is overexpressed in that particular cell, it's just there to take over, give 2 electrons, and recover respiration and recover mitochondrial function. That's associated with the respiratory function. So this is my refractory version of the metformin binding, right? Metformin can bind to complex 1, but it can't bind to NDI1, and NDI1 in part can regenerate NAD and pass 2 electrons. So the first experiment we did this was in cancer, where what we did is we took a sensitive cancer cell lines, and this one is HCT116P53 null. As you can see, in drinking water, metformin reduced tumor growth, but NDI1 rescued this, and we saw this in a variety of cancer cells, and many people have been able to repeat this particular experiment. So the overexpression of NDI1 was sufficient to abrogate the effects of metformin, which suggested that it was intrinsic to mitochondrial complex 1 activity inhibition. People always ask me, why, if you inhibit respiratory chain, why do cancer cells not grow in vivo? This is our work orthogonally using genetics, where we've knocked out all these components of the electron transport chain, including complex 1. What happens is that the electron transport chain is coupled to the TCA cycle, and the TCA cycle is the anabolic engine of growth. So if metformin can get into that cancer cell, it'll inhibit complex 1, the TCA cycle doesn't work, and boom, you don't have the metabolites for growth, okay? Now, does this happen in humans? Well, the closest we've gotten to are these 2 studies, and there's a few others where Ernst Langel and Joseph Lecosel, they gave ovarian cancer patients metformin, and then they took biopsies before and after, and what they found was the TCA cycle intermediates were down in those patient biopsies from these ovarian cancer patients, as was metformin enriched. Another study from Adrian Harris's group, where then they gave metformin, what they could see is an increase in FDG PET in a subset of patients, all right? And that FDG PET uptake of glucose is going to be important in a second. You'll see why. But again, if you inhibit mitochondrial complex 1, typically you uptake a lot of glucose, and that can be measured by FDG PET. Here's the problem with the cancer studies. Largely, they have failed in stringent phase 2 and phase 3 clinical trials. The largest one was a phase 3 clinical trial, and it failed, right, for breast cancer. And why did it? And in our own studies, and many people who work with metformin, you have cell lines at 50 micromolar that are very sensitive to cell growth, cancer cell lines, and others are at 50 millimolar, they laugh at it. Why is that? So what we did is we took an unbiased approach. We took one of those 50, 100 micromolar sensitive cancer cell lines. We did a CRISPR screen only in the metabolic library and simply asked what gene, and if you're sensitive, what is the top hit if you lost that gene? Not very informative, but informative. The OCT3 transporter, the plasma membrane metformin transporter. So this is a sensitive cell line, and that dies with metformin. But in an unbiased screen, if you lost this transporter, then guess what? You lived, right? And I think it'll never happen again, but if you could go back in time and do a breast cancer or prostate or whatever one that you want to do with metformin, if you use at least the transporter as a mechanism to stratify, right? I always say you would never give Herceptin to a HER2 negative breast cancer patient. You give it to a HER2 positive. Maybe using the organic cation transporters and other transporters that are needed for metformin to get in, if you stratified your patients based on that, would metformin work as an anti-cancer agent? Okay, so the transporters matter. So one of the other things we did is Colleen Rezek hooked up with Greg McElroy, and they generated a mouse model where they overexpressed NDI1 in a mouse. As in the way they did it, they put it in the Rosa locus with LOX-P sites around a stop codon. This way you can inducibly express NDI1 in any tissue you want. Okay? Right? So this is what the mouse would look like. You would have the endogenous complex 1 still around, and in wherever your Cre recombinase is active, heart, muscle, pancreas, gut, NDI1 gets expressed. And in that particular cell, you have a complex 1 around and an NDI1. You have both of them around, and we can talk about the caveats that go with it. And now, you basically, what we did, is Colleen did, is she took beta-actin Cre. It comes out in blastocysts at day 4, early in gestation. And therefore, NDI1 would be expressed throughout every cell type. It turns out you can't pass on NDI1. You have to always cross it every time, and there's a selection against passing it on. It's quite interesting. But nevertheless, we can get, with this strategy, NDI1 expressed in many tissues, as in the mice get to adulthood and they look fine, as far as we can tell, by a variety of measures. But the idea here would be you give metformin orally, and if NDI1 is in some tissue, in all these tissues, you could do the classic glucose tolerance test. And as you can see, metformin lowers the glucose with NDI1, and it largely, under standard diet conditions, rescues it. It isn't a full rescue, and we can talk about that in a second. Okay? But nevertheless, the presence of NDI1 makes them refractory. We can do this in a high-fat diet as well, right? So high-fat diet, you see the lowering of the glucose with NDI1, it remarkably brings it back. Not all the way, and I'll talk about that, potentially why. And what this really tells us is something very simple, that metformin, and we think, can hit mitochondrial complex 1 and to lower blood glucose levels. Because we used beta-actin Cre, it doesn't tell us where, right? It could be in any one of these tissues. The liver, if you're liver-centric, the gut, if you're gut-centric, or the kidney, the 3 major places it goes, or maybe the immune cells as well, right? Okay, so naturally the next question was, we could start to generate tissue-specific NDI1. Remember, the NDI1 was in the Rosa locus, and wherever the Cre is positive, if you have a beta-actin Cre, it's everywhere. If you have an ALB Cre, you can do it in the liver. If you have a villain Cre, you can do it in the intestines, right? Just to remind you, the reason why we do everything oral, as this particular study and others have pointed out, that the oral, if you just image where the biodistribution of metformin goes, it accumulates the highest in the gut, then it goes to the liver, and then eventually the kidney, and you excrete it, right? But the gut does accumulate. I think this is not my own data, obviously, but this is quite well known. The gut has quite a high level, up to 1 millimolar plus. It can accumulate and then eventually to much lower concentrations in the liver. And then we'll get to that in a second. The other study, again, this is not our work, but just this paper in PNAS, which shows that FGG PET, if you look at it, the gut is where you get a signal. So this is diabetics, controlled diabetics and diabetics with metformin. You see this increase in FGG PET. I'm told by the oncologist if you have cancer, or God forbid, and you're taking metformin, they tell you to be off metformin for about 2 weeks so you don't get any interference potentially. Okay, can we see this in mice? And we can do the exact same experiment, and where we can do this FDG PET, and you can see it nicely light up. And one idea, of course, is that if you're hitting mitochondrial complex 1, and one of the first things you do is you increase glycolysis and you uptake glucose. So this increase would be compatible with the mechanism of complex 1 inhibition. But of course, you can imagine it could be complex 1 independent, and maybe somehow metformin is hitting the glucose transporters, AMPK directly, or something else. But here, we can actually test as to whether metformin inhibition of complex 1 is necessary for this glucose uptake. Because now we're going to put, using Villancre, NDI1 only in the intestines, right? And if you can see, the data is almost night and day here, where metformin with Villancre, the control, you can see this light up in the colon and the intestines, and it's largely abrogated with the overexpression of NDI1, implying that metformin must be hitting mitochondrial complex 1, right? So we've been paying attention to other people's data coming out about what's happening with metformin in the gut. One of the things from Lydia Lynch and John Long's work is that metformin can increase lactate transiently, and some of that lactate is buffered by an enzymatic reaction that John Long found where you can make lac-Phe. Lydia Lynch can actually show in people who take metformin, you can see this lac-Phe. And one of the nice things in John's paper was they knocked out this particular enzyme specific in the gut with Villancre, and they can show that the lac-Phe levels go down. Well, we would argue that this makes sense with our model because mitochondrial complex 1 inhibition increases glucose uptake. Most of that becomes lactate, and maybe that becomes lac-Phe. And here you go. Here's the control lac-Phe increase, exactly what was reported by those papers in mice. With NDI1 overexpression, only in the gut, you can markedly reduce those levels of lac-Phe. Again, suggesting that the mitochondrial complex 1 inhibition not only increases glucose uptake, but it helps generate the lac-Phe. The other one that's come out of the gut literature is the fact that GDF15, again, I'm not going to get into controversies whether these things have anything to do with body weight or glucose homeostasis. To me, they're great papers because they're nice outputs of what the gut is doing with metformin, right? And so here's the thing where metformin is known through the integrated stress response to increase GDF15. And again, we could ask whether metformin accumulation in the gut by inhibiting complex 1 and is driving that GDF15. So here's the GDF15 in the serum. We can measure them in the jejunum as well. And you can see it's markedly lower when you overexpress NDI1. Again, suggesting mitochondrial complex 1, it inhibits it, increases GDF15, increases lac-Phe, increases glucose uptake. The other thing, and this is really Zach's work, all this intestinal work, it's unpublished, but one of the things you can do is you can just fast the animals and give them a certain amount of food, control mice and NDI1 mice, and look at sort of a postprandial glucose spike. As you can see, it's abrogated. The spike of glucose is abrogated with metformin because the glucose uptake is in the intestine, and NDI1 abrogates that. You can see metformin lowers the insulin levels as well. Again, this is abrogated by having NDI1. The idea being metformin hitting complex 1 sucks that glucose like a glucose sink, very similar to how insulin would do it in the muscle. And this is how we think it might be lowering the glucose levels. You can start to do at 100 mg per kg. We don't, at least in our B6 mice, below this, we don't get a robust glucose reduction in our glucose tolerance test. But you can again see metformin lowers it. With the NDI1, it's largely abrogated. You could do this at 200 milligrams, same thing. largely down with metformin and somewhat rescued by NDI-1. You could do this with a high-fat diet. It begins to, the NDI-1 begins to abrogate it. So in all of these cases, what we start to see is basically this model where we think that metformin inhibition of mitochondrial complex 1 in the intestine and then basically causes the glucose transporters to suck up the glucose, as I showed you with the FDG PET, and this is a mechanism of lowering the glucose. Obviously, the intestine as a glucose sink model has been proposed by others. We would argue, on top of what people have proposed clinically as well as in mouse models, that perhaps mitochondrial complex 1 is the reason why you're getting this glucose uptake. Now, metformin gets to millimolar in the intestine. That is compatible with the mechanism of action of inhibiting complex 1. By the time it gets into the liver, as many people have rightfully pointed out, it's around 50 micromolar, maybe 100 micromolar. Therefore, it may not hit complex 1. And I agree with that. I think the gut data makes sense to us. It's not completely clear whether metformin's effect for hepatic gluconeogenesis would go through complex 1. And we're doing those experiments, but I'm open to other targets. Our data suggest. The other thing is, we have both systems. So now we're doing the harder experiment where we're getting rid of endogenous complex 1, that 45 subunit complex 1, and only having NDI-1 to really clean up the data to make sure how much of it is all going through complex 1 in the intestine, or maybe perhaps. In my last two slides, I just want to show real quick, many of you might be interested in fenformin, another biguanide, very similar effects. Right? NDI-1 abrogates largely the effect, again, in the intestine-driven Cree. The other one is berberine. I didn't know a lot of people apparently use this as a supplement, perhaps these days more than others. You have to give an inhibitor. Thank you for laughing. An inhibitor that allows the berberine to accumulate. And again, in the intestine, it's the highest concentration. And you can see the lowering of the glucose is abrogated again. This is structurally not a biguanide. This is a different molecule. One other thing, people always ask me, should I take metformin? I'm metabolically healthy. Two studies. One is a recent study on clonal hematopoiesis. Apparently, we all undergo a certain amount of baseline clonal hematopoiesis. If it gets to a certain level, these rogue clones can be pro-inflammatory. In mouse models of this clonal hematopoiesis, metformin lowered the expansion of these clones. Notably, in UK biobanks, when they controlled for a variety of factors, it also lowered the prevalence of a particular type of clonal hematopoiesis. And again, clonal hematopoiesis is linked with inflammation, as our last speaker. And I have 37 seconds left. Real quick, the other reason is pollution. You know, Chicago used to be a really polluted city. It's remarkably changed its pollution in the last 30 years because we have more trees than any big city. Unfortunately, the wildfires have made us polluted again. We've gone from 20, 30 to almost up to 150, 200 on that scale. My friend Scott Buttinger and Gokhan Mutlu, they put Beijing level pollution inhalation in mice. They gave it metformin. IL-6 is the thing that spikes with pollution in humans and in mice, and that was lowered. It would be interesting for someone to do a pollution study during the wildfire season and see if my IL-6 levels go down or not. Anyways, I'll leave you with this thought. Not all the biology will be explained by complex 1, but we think some of the pleiotropic actions of metformin that people have reported could be explained by hitting this reversible mitochondrial complex 1 inhibition. Thank you.
Thanks very much, Nav. So next up we have Mark Herman, who's the E.O. Wagner Chair of Internal Medicine at Baylor College of Medicine. And Mark is going to talk about metformin and fructose metabolism.
All right. Thank you to the organizers. Thank you, Lawrence, for inviting me to present at this session. My lab is really not metformin focused, much like our first speaker, and in contrast with Nav. But through kind of a circuitous route, we found ourselves interested in metformin action, possibly as a readout of some of the biology we were studying. And I'm just going to summarize my conclusions at the very beginning, which is that our data, I think, is very concordant with what NAV has shown in terms of complex 1 and mitochondrial function, particularly in the intestine being a major target for metformin and its metabolic effects. And I'll get into that towards the end of this talk. But my laboratory really focuses on fructose metabolism, and this is just a cartoon highlighting the fact that as opposed to glucose, which is the circulating sugar in animals, in plants, sucrose, right, is the circulating sugar. It's the vascular sugar, and this is kind of the base of our food web, created through photosynthesis and really allows life as we know it to exist on this planet, right? Sucrose is a disaccharide composed of one glucose monosaccharide and one fructose monosaccharide. And whereas glucose is the primary energetic and synthetic fuel for most tissues and cell types throughout Animalia, it circulates at 5 millimolar concentrations, as this audience well knows. Fructose is not a major source of energy or synthetic fuel for most of the cells or tissues in the body. And in peripheral circulation, it's only found at about 10 micromolar concentration, right? But it's present in sucrose, this dietary sugar, in equal amounts to glucose. And so fructose is catabolized in the body much differently than glucose is. And it was recognized beginning really in the 1960s. This is a paper from the late 80s from Gerald Reaven, who was using models of very high fructose in diets to show that a single dietary nutrient could, when given in great excess to what is typically consumed, could recapitulate all of the features of metabolic syndrome. So this was a rat model where he was giving 60% fructose diets, right? So the average fructose intake in Western population is about 7%. So this exceeds that by tenfold. It's not physiological by any means. But if you give a 60% fructose diet, you can cause all of the features of metabolic syndrome within a matter of weeks in animals. And that's true for many of the phenotypes, particularly lipid phenotypes in humans as well. And it was actually this model that he used to codify the idea of syndrome X, which became metabolic syndrome or cardiometabolic disease, that a single dietary nutrient could cause all of these features. And so I've mentioned that fructose is metabolized differently than glucose, right? So when you give an oral bolus of glucose, like in a glucose tolerance test, right, the reason that is an effective test is that very little of that glucose is actually metabolized first pass in the intestine or the liver. Most of that ingested glucose in an oral glucose tolerance makes it past the intestine and the liver, right? Stimulates insulin secretion, which is required for its disposal. And it's that disposal, insulin-mediated disposal of glucose, which makes a glucose tolerance test useful as an index of both insulin secretion and peripheral insulin sensitivity. But fructose is completely different, right? If you give an oral bolus of fructose, the vast majority of that is metabolized in the gut first pass. So there's a series of nice studies beginning in the 1970s and most recently, about 5 years ago, that if you give stably labeled fructose to humans and you, as an oral bolus and use some complicated tracers, you can measure the clearance of fructose first pass. And only about 14% of an orally ingested 30-gram fructose load makes it past the gut first pass. And so this results in some interesting observations, which is that if you measure fructose in the portal circulation after an oral fructose load, you can measure about millimolar concentrations. And at the same time, you can only measure about 10 micromolar concentrations, 100 micromolar concentrations in peripheral circulation. So the combination of the intestine and the liver really is efficient at extracting that ingested fructose and metabolizing it. And so there is an enzyme, ketohexokinase. There's a couple isoforms of this, but the high activity isoform is really only expressed at high levels in two tissues. That's in enterocytes and in hepatocytes. And this is the key enzyme for phosphorylation and metabolism of fructose. And so when you ingest fructose, some portion of this is taken up in the enterocytes and metabolized within the enterocytes themselves. The portion that makes it past the enterocytes into the portal circulation of the liver takes up a large portion of that fructose and phosphorylates it to fructose 1-phosphate, where it then enters the triose phosphate pool along with glycolytic intermediates. So there is a really nice experiment of nature showing that this fructose metabolism in either the liver or the intestine is actually important for fructose-induced metabolic disease. And that is there is a condition called benign essential fructosuria. So these are people that have inactivating mutations in ketohexokinase, and they have no metabolic problems. It's not called a disease. It's called benign essential fructosuria because these people have they do not come to clinical attention. They've all been identified by accident, the cases that have been reported, because they do not have any overt clinically significant syndrome. But these individuals, if you give them a 30-gram bolus of fructose, you can measure millimolar circulating fructose in the peripheral circulation. So it's not fructose per se that causes the metabolic disease. It's fructose metabolism that is problematic. And this has actually been reproduced in genetic mouse models. You can knock out ketohexokinase, and those mice have no metabolic problems. And when you challenge them with high fructose diets, they do generally well. They don't become obese. They don't develop dyslipidemia or insulin resistance. So fructose metabolism is essential for fructose-induced metabolic disease. Now, I'm going to summarize. This is like the last 10 years of work in my laboratory and really what we focus on. I'm going to show it in one slide, which is we focus on a transcription factor, something called carbohydrate responsive element binding protein. It's a transcription factor that senses intracellular glycolytic intermediates. It's activated by these intermediates to regulate genomic programs that regulate global metabolic programs. And we've shown that though this factor was discovered based on its ability to be activated by glucose and glycolytic intermediates in cell culture, in vivo, in humans, and in animals, in the intestine and the liver, this is robustly activated by fructose ingestion. And I can go through the biochemistry of why that's the case. Turns out it's common intermediates to glucose and fructose that activate this. It's just that because fructose is metabolized so efficiently in the enterocytes and the the hepatocytes, you get robust activation of this transcription factor. But we've done a lot of studies knocking this out of the liver and the intestine. And to summarize again, a decade of work, CHB activation in the liver is essential for fructose-induced metabolic disease, but CHB in the intestine is essential for dietary fructose absorption. And that is because, and we'll get to this when we get to metformin in a little bit, which is that in the intestine, CHB regulates expression of a transporter called GLUT5, which is the sole intestinal lumen fructose transporter. So it is the only thing that your intestine expresses that allows absorption of fructose across the intestinal membrane. And if you knock out CHREBP or if you knock out GLUT5, if you knock out CHB, GLUT5 expression goes down. The same thing happens if you knock out GLUT5 itself. If you challenge those animals with fructose, they develop severe malabsorption syndrome, basically stop eating because they can't absorb the fructose and get microbiome overgrowth and dysbiosis and do very poorly. While we were doing this work, there's a really nice paper from Chulsung Zhang and Josh Rabinowitz, who also began studying fructose metabolism with a focus on the intestine. And they showed very nicely that when in this case it was C57 mice given a large bolus of fructose, the vast majority of that fructose, again, was metabolized within the enterocyte, and a large portion of that fructose within the enterocyte was converted to glucose and put back into the portal vein as glucose. So it highlighted something that's been known for a long time but not really well appreciated, which is that the intestine is a gluconeogenic organ. It expresses glucose 6-phosphatase levels at very high levels, comparable to those in the hepatocyte, and that there is some degree of basal glucose production in the intestine at all times using a variety of different substrates, a lot of amino acids. But that fructose is preferentially converted into glucose in the intestine and put back into the portal vein. This is a very nice paper where they demonstrated that. And we had been doing this work on fructose metabolism and studying its effects, its role in developing metabolic disease. And we had noticed in our studies accidentally that different strains of mice had markedly different responses to fructose. So these are two mice. The left is the C57 Black 6, which is the mouse used in the majority of metabolic studies around the world. And that's in part because it was the first mouse sequenced and the first mouse where genetic mouse models were generated. So most of the lock-stop LOX alleles and the CRE alleles are generated on the C57 background. But the mouse on the right is a C3H-HEJ mouse. And we noticed accidentally early on in some studies that the C3H mice seem to be very sensitive to fructose-induced disease, whereas the C57 mice are largely resistant. And this is published data. This is just taking C57 mice versus C3H mice. These are wild-type mice purchased from Jackson, put on a 60% fructose diet. I think this was a 10-week time point where this data is shown. And you can see that the C57 mice on fructose, they haven't gained any weight. In fact, they've not gained as much weight as the C57 mice on chow, whereas C3H mice gain a tremendous amount of weight on a high fructose diet. And the C3H mice develop hyperinsulinemia and hypertriglyceridemia and severe hepatic steatosis, and much more so than the C57 animals. So there's more strain-dependent differences in fructose sensitivity. And this is something we've been studying for a while. And it led us to do some studies. Again, we speculated, in part on data from the Rabinowitz lab, in part on our own lab, showing that CHRB in the liver was critical for fructose-induced disease. Well, maybe there's a difference in how fructose reaches the liver between these two strains. And so we started doing tracer experiments using UC13 fructose, gavaging mice of different strains with UC13 fructose, and measuring the fructose absorption in the portal vein and measuring its conversion to glucose in the portal vein. And this is some of that data. So this is C57 mice gavaged with fructose and the portal fructose levels. And this is C3H mice gavaged with fructose, and this is the portal fructose levels. And this is M3 and M6 glucose in the portal vein after fructose gavage. So this is glucose in the portal vein generated by gluconeogenesis in the intestine shortly after fructose gavage. And you can see the C3H mice are much more efficient at producing glucose than the C57 mice. Okay, so this is a demonstration of two things. That fructose absorption can be markedly different between strains of mice, and that can result in differences in glucose production as an index of metabolism in those intestines. And so we went on to prove that show that if we knock out if we repeat this on GLUT5 heterozygous animals. So this is GLUT5 is the transporter again for fructose in the intestinal lumen. We can reduce portal fructose levels by half in both the C57 and in a mixed background, and also reduce the glucose production from that labeled fructose by half. All right, so this is really dependent on the rate of fructose absorption in the intestine. And this is one of those things when you start to do any kind of omics, we're starting to get into metabolomics to make these measurements and to measure tracers, is that you don't just measure the thing you're interested in. You end up getting lots of data on lots of different metabolites and run into observations that are completely unexpected. And this was one of those fortunate circumstances. So these assays were being done by the Wenxing Tong, who was a graduate student in my lab at the time. And fortunate she's here today. She's moved on to bigger and better things than working in my lab, and I'm happy for her. But she did really some fantastic work when she was in my laboratory. And she observed that in some of our studies where we were measuring portal metabolites and peripheral metabolites from individual animals sampled at the same time, that the level of TCA cycle metabolites was much higher in the portal circulation than in the peripheral circulation. And that was very surprising to us. We didn't really have an idea of why portal TCA cycle metabolites should be elevated compared to peripheral circulation. But one of the obvious hypotheses when you encounter data like that is, well, maybe these TCA cycle metabolites are being made in the intestine, whether it's the microbiome or the enterocytes, and being secreted into portal circulation, and maybe they're then scavenged in the liver. And I'm not going to go through all this data. It's published a couple of years ago, but it turns out that enterocytes are a major source of circulating TCA cycle metabolites, both in the portal and peripheral circulation. We've shown that now using fructose, UC13 labeled fructose as a tracer where we can quickly label, increase labeling of, this is UC13 fructose gavage into animals, and measuring labeled succinate in the portal circulation. And you can see within 10 minutes, we're getting substantial labeling in portal succinate. It's comparable to what's in the intestinal wall, and the intestinal tissue, and we don't see that labeling in cecal content. So this is not coming from the microbiome. This is actually enterocytes producing this. And Wenxing did some really phenomenal isotope tracing to really demonstrate that, and it's in this paper. But you can see that by 120 minutes, you have about 30% of all portal TCA cycle metabolites are labeled from fructose. And so it's really substantial labeling of this circulating TCA cycle pool from fructose, which is being metabolized selectively in the enterocytes. So, this led to a couple questions, and I'm gonna tell you up front that I don't have the answer to most of these questions. These are things we're still interested in, and we don't really know. But, what are the normal substrates for intestinal TCA cycle production? Do intestinally derived TCA cycle metabolites serve as signals of intestinal metabolic state and or nutritional signals to the liver and the other tissues? These are things we're very interested in. But this third point is something we could address quickly, and what brings me here to presenting at this forum, which is, can we use this biology as a tool to probe intestinal metabolism in live conscious animals and possibly in the future humans, right? So this gets us to metformin, and we are aware of NAV's excellent work and the accumulating evidence that intestine may be a major target of metformin. And we wanted to know whether we could use this TCA cycle, this fructose paradigm and TCA cycle biology to begin to assess in vivo, in animals, is metformin likely impacting the metabolic function of intestinal tissue? Now, you've seen data that NAV showed that there's good evidence at this point that metformin increases glucose uptake in the intestine through FDG PET scans, but we were interested if there were other mechanisms as well. And so this is a paradigm that we generated a cohort of obese mice with a high fat, high sucrose diet, and then once they were obese, we put them on 3 weeks of metformin, oral metformin at a dose of 50 milligrams per kilogram, which is a dose that's comparable, or what we think is comparable to what is achieved in humans. And you can see that this did not result in any decrease in body weight over the 3 weeks they were on this. And fasting glucose tended to be a little bit lower in the metformin-treated mice, but did not achieve statistical significance. But they were definitely glucose tolerant, but during a glucose tolerance test, and you can see the area under the curve here. So metformin, 3 weeks of metformin produced glucose tolerance in these mice. And then we used our UC13 fructose paradigm and began to ask, was metformin impacting mitochondrial function in the intestines of these mice? And that was definitely true. So you can see this is labeling from our UC13 fructose in portal succinate and the water-treated versus metformin-gavaged mice. And there is a reduction by about half in this labeling in the metformin-treated animals. And you can see that for succinate, you can see it for malate, you can see it for citrate, and it was the case for every TCA cycle metabolite we measured. And along with that, you could detect that there was actually a decrease in portal glucose levels while systemic glucose levels were still normal. So there was a selective impact on glycemia in the portal vein in these animals that you could not detect in peripheral circulation. And so it seems that this could be a combination of, and then I guess the last piece I want to show you is that this is labeling of the glucose in the portal vein. So metformin decreased M0, the unlabeled glucose, but it also decreased M+3, labeled glucose. So there was a reduction in the conversion of fructose to labeled glucose in metformin-treated animals, suggesting that we've impacted or inhibited intestinal glucose production in these animals. Okay, so this is just to summarize. I'm over 5 seconds. So, all right, pretty good timing. So enterocytes are a major source of circulating TCA cycle metabolites. Orally delivered fructose rapidly labels the enterocyte and circulating TCA cycle metabolite pools. An analysis of this labeling pattern of circulating TCA cycle metabolites intermediates after oral consumption of labeled fructose may be useful to assess intestinal metabolic state in live conscious animals. And we haven't done this yet, but we're hoping to do this in people as well. So metformin impairs intestinal fructose metabolism and intestinal glucose production. And we believe that, we haven't proven this by any means, but we believe that the reduction in glucose production, gluconeogenesis, has been detected, throughout decades in metformin in people. A lot of that reduction in glucose production may actually be in the intestine and possibly the liver also, but we don't know which tissue contributes to what degree. So I'll leave you with that. All of the people that contribute a decade of work here.
Okay, so please join me in thanking all the speakers for their great talks. Thank you.